Welcome back, BIO178. In the next installment here, we will focus on the conclusion section of your poster. So here, if we go back to that template example, we are focusing now on this section here on the far right. So overall, in the conclusion section, what you're doing is thinking about what your results mean more broadly in a broader context. You want to think about why they're important, why other people should care about them, and then you also want to think about some extensions and potential future work that you can do to gain some additional insight to build on the results that you obtained in your particular study. So first of all, we will break down what specific things you should be including in this particular section of your poster. So again, what you want to make sure you're doing is describing what your results mean. So this little weird meme thing is shrugging its shoulders and saying, so what? So that's essentially what you want to explain to people in this section. You want to say why these results matter and why they should care about them. So in the results section, you're not just restating the results. That's already been described in the results section itself. But what you're doing here in the conclusion section is describing what your results actually mean. So why they are relevant and what the broader implications or applications of those results are. So an example of that might be something like this. So this is pulled from that example template I gave you. This is one of the excerpts I included in that particular conclusion section. And what it says is the identification of outlier loci, so that was what this poster was focused on, could be indicative of adaptive genetic variation. So here, this is what's in bold is taking the results a step further. So the results themselves are basically, we identified some outlier loci, but when you're talking about the so what portion, that's what's here in the green, what I've underlined in green. So we're thinking about, we've identified loci, that means there's probably adaptive genetic variation going on. You could then expand on that and say we care about that because these different populations are exposed to different temperatures. They are likely going to fare differently with climate change and increasing temperatures, etc. So this is an example of describing what your results mean in a broader context. You also want to make sure you're describing how your results compare to other studies. And an example of that, to illustrate, we will go back to something I brought up many weeks ago at this point, at the beginning of the semester, where I was describing these two different contrasting results from different papers, right? So in this first bullet point here, I have a description that says for this particular snail, there is no structure from Oregon to Southern California. However, in my own paper, in my own research, I did indeed find evidence for regional differentiation. So this is an example of how you want to describe how your results compare to other studies. So in this case, if I were creating a poster based on my results of identified outlier loci, I would mention that my results are in contrast. They do not agree with this previous work. And if that is the case, you want to make sure you're describing why you think that is. So in this case, as we've talked about previously, there are a couple different reasons why my results didn't quite match previous results. One of the potential reasons is we use different molecular techniques. So I used high throughput sequencing. This other study used single mitochondrial markers. I also was focusing on adaptive loci, whereas this previous study was using neutral loci. So you're comparing your results to other studies, and again, if they don't match, make sure you're describing or thinking about why that might be. In your conclusion section, as I have briefly mentioned, you also want to make sure you're describing future work and next research steps you might take. So for an example of this is again another excerpt I pulled from that template conclusion section that I provided for you. Again, that poster, the main finding was identifying outlier loci. So we've talked about that several times in lecture, but that poster that I've given you as an example was actually the first time I presented those results. So they were still in the preliminary stages, but I knew I had some evidence of identifying outlier loci. So this numbered bullet point I have here is describing future work I propose, next steps I propose to take based on the fact that I had identified outlier loci. So what I have written here is that phylogenomic trees will be constructed and patterns of phylogeny between trees constructed from neutral versus outlier loci will be compared. So again here, what I've done on that poster is identify outlier loci and then describe what I want to do next with that. And that can be creating these different trees based on these different markers. And in fact, I did do that, and we've talked about that in lecture. So as a refresher, these are the two different SNP trees that I created. On the left is from the neutral loci, on the right is from the outlier loci. Again, when you're using the outlier loci, you can start to see these patterns where you see clusters of just northern individuals or just southern individuals. So this is an example of how you think about your data and based on the things we've learned in our class, what different next molecular ecology related steps you could take to build on your results and build on your analysis to gain even further insight into the topic and the research question that you're investigating. So as an example, here is that section I pulled from my poster. So we're addressing both conclusions and future work here in this section. And when I put these sections on my poster, I generally put them as either numbers or bullet points. And that's just to help so we're not just having this huge block of text, but it makes it a little bit easier for the reader or the audience member to makes it more visually appealing. Okay, so we know those are the things we want to include in our conclusion section. How do we go about doing that? The first thing I recommend doing, if you haven't done it already, so a literature search is actually, you can do something at the very beginning of your project if you haven't done it already, but it's especially important for the conclusion section. So what you want to do is do a literature search for related studies. Two good search engines to do that are either Google Scholar and or Web of Science. And when I say do a literature search for related studies, let me explain what I mean by related. So here, let's use the example of the DD-RAD study with C. funebralis, which is what I have described as examples throughout, and that's also the example template poster that I gave you from my previous work. So there are several different categories I think about related studies falling into. So if the study that I did was looking at DD-RAD techniques in C. funebralis, one category of related studies might be, has anyone else used DD-RAD in this same species? So DD-RAD in C. funebralis, but maybe in different populations or different geographic sites. That's one example of a related study. Another category for a related study might be using the same kind of DD-RAD techniques, but maybe instead of in C. funebralis, it's in a related species. So maybe it's in another intertidal snail or another marine mollusk or something like that. That would also be considered a related study. And a third category might be where it's not using the exact same technique, but you're still using some kind of genetic molecular marker. So that other example, when I compared my results to the previous results, they weren't using the same technique. They didn't use DD-RAD, but they were still using genetic molecular marker to compare population differentiation. So that's considered a related study because it's using a similar molecular technique in the same species. So here, to summarize, again, when I'm thinking about looking for literature that's related to my study, it can be same molecular technique in the same species but different populations. It can be the same molecular technique in related species, or it can be a related molecular technique in the same species. And when you're looking for these related studies, you want to pay attention to a couple things in particular. The first thing you want to pay attention to is how your results compare to the findings of these previous papers, again, as we've already talked about. And to find this information, you want to make sure you're focusing on the results section of those other papers. That's where they're going to describe, of course, the results that they found, and you can compare what you found in your study to their results. Another thing you want to pay attention to when you're reading these different related papers is what next steps they proposed in their studies. So that's something that you're doing in your work, right? In this conclusions and future work section, you're proposing next steps to take. So a nice thing to do is see, okay, what did they propose in these other similar studies? That might give me some ideas for my project. And so if you're looking for the next steps that were proposed in these other studies, here you want to look in particular at the discussion section. So that's where in scientific papers, they're putting their results in a broader context, describing any potential caveats of their results, and then also thinking about future work that can take the next step to build on their topic and provide more insight. And so once you've done that literature search, you have an idea of how your results compare to their studies, and you have some new ideas for future work you can propose based on your group's results. Then you just want to, just like this cat at the laptop, which I've been using for all of these videos, you want to write your corresponding text for this conclusion and future work section. In general here, you want to include about 3 to 5 bullet points, so you can do either, again, bullet points, or you can number them, but I definitely suggest breaking it up that way so it's not just a thick wall of text. And for those 3 to 5 bullet points, make sure you're describing the broader impacts of your work, why people should care, and then also at least 1 bullet point to address future directions that this work could take.